So, good afternoon, everybody, and welcome to this event with InDex Pharmaceuticals. With me today, I have the CEO, Jenny Sundqvist, and the CDO, Eva Arlander. Just some practical info for the audience. You're more than welcome to post your questions, and I will hand them over to management during the presentation. The first part of the presentation, Jenny will carry out, and after that, Eva will dive a little into the phase 3 study and the latest PK study. So with that said, welcome Jenny and Eva.
Thank you very much. A pleasure to be here. So starting out with our forward-looking statement, don't need to read it all. You have seen it before many times, but important to show. So today, as Klaus said, it will be myself, CEO, presenting on InDex and our current status. And then Dr. Eva Arlander, our Chief Development Officer, who is in charge of all our clinical trial programs, will do a bit of a deep dive into our PK study results, which we reported it and sent a press release out on March 15th around those results. So that's the plan for today. So starting out with a bit of background, we are a Swedish biotech company based in Stockholm with origins from the Karolinska Institute. We have cobitolimod being developed for ulcerative colitis, and we are currently in phase 3 with that compound. We also have a quite broad portfolio of preclinical stage assets from the DIMS platform, but that's not the focus for today. We're listed on Nasdaq First North, the ticker is InDex, and our main shareholders, the biggest ones are Link with 13%, HBM, and Fjärde AP-fonden with around 10% each, and then Handelsbanken Fonder. And then a large number of retail owners. So our team, quite flexible, highly knowledgeable, as biotech industries are. We have a board of directors consisting of 6 persons with extensive, long experience in the pharmaceutical industry. And we also have expertise in the finance sector specifically. The team in the InDex organization is one of 18 people. We have a mix of full-time employees and then highly specialized consultants who are part-time or on an as-needed basis. An extremely highly competent team. It's diverse, 67% women, both actually in the company and in the board. We have team members of 7 different national origins, which is a pleasure, and a third have PhDs in the relevant inflammatory area where we're currently active with cobitolimod. So where are we with our main asset? We are currently in phase 3, as I said, and we are targeting left-sided ulcerative colitis. We have currently started induction study number 1 and the corresponding part of the maintenance study. So the patients from induction study 1 then flow into a maintenance study, and that's what we currently have started. There's going to need to be an induction study number 2, which is at the bottom in sort of a fainter color because it's not started yet. Regulatory authorities do require 2 studies to apply for market approval. So that's why we have this set up. And it's quite an industry standard that you have 2 induction studies that flow into the same maintenance study. So induction study 2 and its corresponding part of the maintenance is not funded at this time, but the plan is when we have our top line data from induction study 1, that's when we'll press the go ahead button and raise money and start study number 2. So our study status, we're currently approved in all planned countries, 30 of them. So we have regulatory and ethical approval everywhere. We have had a bit of a slower start than expected, and that was communicated at the end of January. And I've covered all the reasons for that. I'll take any questions if there's more after this. We have set in action an action plan to actually progress on the speed and catch up and ensure that we proceed from now on at a greater pace. And we expect to present the outcome from the dose selection milestone in Q4 this year. And I'm going to go into in the coming slides exactly what that dose selection milestone is and what it means. In Q4, I will also come out and comment on the new overall development timeline, including when we expect top-line results for induction study 1. So our next milestone, as we said, is a blinded dose selection. And what is that? Blinded means, of course, that we don't know, we can't see the data, the treating physicians don't know what compound they're given, the patients don't know what they're given. And that's a requirement, actually, to be able to use a study for regulatory filing. So blinded means that an external committee will actually, the data monitoring committee will go in and look at the data and then give us an answer. So in induction study number 1, we aim to recruit 440 patients into this induction study. They will be randomized into either cobitolimod 500 milligram or 250 milligram or placebo. When 132 patients out of the 440 have been treated for 6 weeks, and we have the results from their treatments, that's when the DMC will go in and look at the data and make recommendations back to us whether we should continue with a 500 milligram dose or the 250. And from that point on, we'll also only continue with a so-called winning dose, depending on what that is. So we will also at that time receive the results of what's called a futility analysis. And that means that if cobitolimod is unlikely to be better than placebo, the study will be stopped. It's a general industry standard. Of course, that's not expected, but you know, if that's the case, it's not ethical to continue clinical trial. So a bit more around what we will know in Q4 this year. What will this blinded dose selection milestone actually give us? So what we will know, sorry, I have a bit of a COVID and cold. Cobitolimod, what we'll know is that cobitolimod has the potential to be better than placebo, because if it was worse, the study would have been stopped. We will also know that we have no alarming safety signals, because if DMC observe alarming safety signals, they will stop the study. And we know that we can proceed with a conclude program as planned. So that's the good news. But I also want to cover what we won't know, because there's some expectations that we will have data that we actually won't receive at this point in time, since it's blinded. We will not be told anything around the magnitude of efficacy. So the DMC committee with external experts, they will be able to look in the data, but they will not share any of that with us, and we won't have access to the data. So we won't know, even if we get the go-ahead to continue the study, we won't know whether the efficacy is benefit is tiny or whether it's huge. We don't know. We can only be happy that we can continue the study. If we get the decision that please continue the study and do so with 500 milligram, we cannot assume that the 500 milligram dose efficacy is much greater than what 250 has shown in previous trials. Because in theory, we could happen that 250 in this study performed worse than placebo and 500 just above. I mean, I'm speculating, but we can't assume that it's much greater than 250. If we get the go-ahead to say, yes, please continue the study, but do it with a 250 milligram dose, we can't assume that efficacy will look similar as to what is done in previous studies. We just know that it has efficacy. And we cannot assume that its efficacy is greater than what the 500 show, because it could, in theory, be that 500 and 250 show the exact same efficacy. And then obviously you want to give the patient as low of a drug load as possible, because it's not ethical to just give them lots of drug for the sake of it. So that also might be the reason. So that's important to know. And that's what we will know at the end of this year in Q4. So at the end of this year, we'll know, yes, you can proceed with the study. Well, hopefully, we're assuming so. We will proceed with X dose, and we won't know any efficacy data per se. So I just wanted to give that very high-level summary update of where we are with a phase 3 program and what our next milestone is before then Eva dives into the results of the PK study, which is another type of study.
So just very briefly, Jenny, and thanks a lot for your introduction. So when we get the top line data, the total top line data, then you start up the second arm of the study. Is that correct?
When we get top line data for induction study 1, so that's when all 440 patients have gone through the study, we analyze the data, and then we actually get the top line efficacy data. So then we will know the results of the primary endpoint and the secondary endpoint.
Yes.
And assuming that's positive, that's when we'll go out and start study 2.
Good. And at that time, you also know, well, you already know whether it's 500 milligram or 250 milligram, but you don't know the efficacy. Let's theoretically say that is the 500 milligram. Does it make sense then to do the second study?
Oh, the second study will only be done with a winning dose. So that's a good question. So at the dose selection at the end of this year, we're going to pick what we call a winning dose. And anything from that moment on will only be done with a winning dose. So the remainder of induction study number 1 and study number 2 that will be started later will only be done with a winning dose.
Good. Okay. And if they are similar, there's no reason to continue.
If they're similar, we would pick the 250 because there's no reason to give patient a double load if you have the same efficacy.
I fully understand. There's two more small questions here before we start with Eva, if I may. How many patients have been treated so far, and why don't you give continuously updates regarding the patients treated?
Yeah, and that's a question that we have gotten before, and that is based on experience from previous studies and the general market or how recruitment for ulcerative colitis works. So in the past, there have been numbers given out. Now we have 50, 100, or that people can follow progress. The challenge with UC is that you don't have a linear progression in recruitment. So you usually in ulcerative colitis have very slow starts. It takes a long time to get clinics on board, but once they are on board and they've recruited the first patient, it's quite a steep increase. So the issues that we and other companies have run into if you let out numbers is that a lot of analysts and investors start making predictions and extrapolate, and you end up with completely wrong expectations around timelines. So that's why we have chosen at this time not to communicate patient numbers, but rather milestones. So our next big milestone will be the dose selection in Q4 this year. And at that time, I'll also come out and be clear about what future milestones do we have and when will we communicate along the study timeline.
Well, that's very good expectation management you do there, Jenny. Thanks a lot. Then the last question here before we hand over to Eva. You mentioned in your previous slide that you have changed a little. You have speeded up the roll in of patients. What have actually changed compared to previously? Could you elaborate a little on that, Jenny?
I can give a few examples. So we have done a number of activities, a long list. I won't bore you with the entire one, but we have, for example, analyzed potential of clinics and have a list of what we call VIP clinics, and we treat those with special attention and care. So they get direct contact from us at InDex with the sponsor directly to them, and get any support and help they want, and they are the high recruiting potential clinics. We have, for example, worked on technical equipment and user guides to make any technical aspects easier. We have call support in terms of data. For example, clinics have questions around firewalls or how do I use this in Wi-Fi, any questions that come up, which provides support with them regarding that. And then we have also put in place, for example, incentive programs for the clinical research associates who are out there recruiting clinics and speaking to them. So that's just a few examples of what we have put in place.
Thanks a lot, Jenny. And with that said, I would like to hand over the word to Eva. Please, Eva, carry on.
Thank you, Klaus. And well, I'm going to talk about the pharmacokinetic study that we have just recently communicated about and give more details to that. And I would like to start with why we did this study. Why do we want to know the pharmacokinetics? And the cobitolimod is a local treatment in the colon. So it's given locally to patients with ulcerative colitis, distal ulcerative colitis, left-sided, you may also call it. And the drug is supposed to exert its efficacy locally. So systemic concentrations are not, they are not dangerous, but they are not really necessary for our treatment to work. But of course, it's important to understand what we have. So what systemic concentrations do we have? So therefore, we did this study. And so then it's, of course, an important part in our regulatory puzzle, you may say. So this is an important piece of our regulatory puzzle, all of the studies that will constitute our final assessment of cobitolimod as a new treatment. So by saying that, what did we do? It was an open label study with cobitolimod, which means that there was no placebo group. So all patients knew that they were given cobitolimod. And we had the highest dose in the phase 3 program, which is then 500 milligram of cobitolimod. The patients that were included in this study had the same severity of ulcerative colitis as in the phase 3 program. It was moderate to severe ulcerative colitis. And it was both men and females who could participate in this study. They had to have an active disease, so they needed to be in the inflammatory stage of their ulcerative colitis when they were included in the study. And that is measured by a score, the 3-component Mayo score. And in my next slide, I will speak a bit more about that. So I leave that for now. But the Mayo score should be between 5 and 9, which is then also similar to what we do in the phase 3 program. And the dose then, again, 500 milligrams. It was the same dose schedule that we have in the phase 3 program. So it's given every 3rd week to these patients. And our primary endpoints in this study were all associated with pharmacokinetics. So it was maximum plasma concentration, called Cmax, and when does the maximum plasma concentration occur, which is then Tmax, sorry. And there you have the area under the curve, which is then a measure of the systemic absorption, and then half-life. And as secondary endpoints, of course, these are always important in clinical studies. It's safety and tolerability. So what is then the 3-component Mayo score? It's a 3-component score, as you can hear by the name, and the 3 parts are then the endoscopy, which is when you look at the patient's colon from the inside. So that can have a score between 0 and 3. And then how many stools does the patient have per day, which then can also have a score between 0 and 3. And then whether or not you actually have rectal bleeding. So the maximum score can be 9, but they had to have at least 5 in this study. And when are you then healed, so to say? When are you in clinical remission? Well, then the rectal bleeding should be 0, and you should have a stool frequency of 0 or 1 and an endoscopic score of 0 or 1. So the study design, these patients were then, they would come to the clinic and they were given the first dose. So first they were endoscopies done and the clinical symptoms were retrieved, and they were given the first dose, which is then week 0 in this one. And then the plasma concentrations were taken. The plasma was taken at specific time points up to 24 hours after the first dose. And then the patients came back after 3 weeks, and there was a health checkup basically, and adverse events, of course, were also collected during the whole study. And at this time point, they were given the second cobitolimod dose, and then at week 6, so 3 weeks further down the line, they came back, and if they were then in remission, so there was then sigmoidoscopy when you look into the colon, and the clinical symptoms were also measured. So if the patient was in clinical remission at week 6, they were given the 3rd dose of cobitolimod. And why we did this was that we wanted to study whether there was any big difference between having a healed colon and having an inflamed colon, whether there would be any change, any difference in the systemic absorption of cobitolimod for this. So yes. So these were then the results, the included patients. So we had 8 patients in this study. 1 patient was excluded from the pharmacokinetic analysis because he went to the toilet just immediately after the dosing, so we didn't know whether or not he actually had any dose given. And of the then 7 patients included, 4 of these achieved clinical remission at week 6. So we could then measure the pharmacokinetics in these 4 patients who healed during the study. And the patient that healed were the ones marked in orange, as you can see in this table. So there was a difference in the Mayo score at baseline. So there was not just patients with only mild symptoms that were improved in this study. Yes. So what did it look like in detail then, the systemic concentrations? So this is a picture of what it looked like in the plasma concentrations when the patients were given cobitolimod in the inflamed state. And as you can see, we say that this was a limited systemic absorption. And what we mean by that is that the patients had concentrations beyond 4 hours. Half of the patients had no concentration in the system. And after 8 hours, no patient had any concentration in the system. And the concentrations were low in comparison to what we see in other studies. I will come back to that. And you can see the details of the data in this table below. So what happened then with the patients who had the healed colon? Well, it looked like this. So you can say that there was a tendency that the patients with a healed colon looks like they had a slightly lower absorption than the ones in the inflamed state, which is, of course, what you could also imagine would happen. There was not a big difference. There was still some variability, not large variability, but this is what it looks like. So beyond 4 hours, no patient had a concentration systemically. And yes, I could just hear myself. That's why I was hesitating. There's an echo. So what do I want to say with this? Well, I would like to sort of put these pharmacokinetic data in the perspective of what other data do we have. Since for cobitolimod, we do not have an intravenous dose in humans, and therefore we cannot speak about the absolute systemic bioavailability that you often mention, but we can put these systemic concentrations in perspective of other data that we have. And for example, then in monkeys, we have a rectal administration in monkeys where we also have intravenous administration, and then thereby we can measure the absolute bioavailability, which is then below 1% in monkeys. And we can also say that the highest Cmax that we obtained in this patient study is far below than the lowest effective concentration that we see in an in vitro model that we use for the pharmacologic activity of cobitolimod, which is then a functional TLR9 in vitro assay. So much below that. So there will not be any pharmacological activity based on the concentrations that we obtained systemically in these patients, if you look at this. And then furthermore, we have a very large safety margin based on the toxicity studies that we have done in the program. So what was our conclusion? Well, we say that we see this limited systemic uptake of 500 milligrams cobitolimod, which we then would sort of strive for, that we have a limited systemic uptake, both for patients with active disease and when they are in clinical remission. And also, of course, then that cobitolimod was well tolerated in this study, very few adverse events, and there were no serious adverse events in this study. So This was my last. No, I have one more slide. What are we going to do with these results then? Well, we are currently writing them up for an abstract at a scientific congress focused on IBD, of course, and then we will also write a scientific article of it. Of course, all the conclude program investigators that we are speaking to frequently will know more about these results. And also our key opinion leaders in both the European and US advisory boards.
Thanks a lot, Eva. So with that said, it's for me it looks like as an outsider it looks like very good efficacy data as well. And I've followed you for quite a while, and I know that the safety issue has actually never really been a case. But now we see some good data with the 500 milligram dose, and you're actually able to use that in some kind of, maybe I used the wrong word, lobbying into the phase 3 study. Is that correct? Or
well, you know, I'm always conservative when it comes to this, so yes. I would say that an open study is an open study. Yes. Of course, these patients were given cobitolimod, but still it's an open study. So as you know, you should never expand too much on efficacy data. What was good for us was that we had these patients in clinical remission, which then allowed us to measure also the systemic availability at remission. So we're happy for that result.
Yeah, so important not to extrapolate on the data, but at least it's a good support for the current study. Just another question, Eva. Did you carry out this study in one clinic, or is it like the phase 3 study in general that you carried out on several clinics?
No, it's one clinic.
In one clinic, okay. Yeah. And that's the procedure you do?
I mean, it's always good to have this type of study in one clinic because it's measuring the pharmacokinetics. You need to be very precise when you take the blood samples, and if you have this spread out on many clinics, it's more challenging. And also, of course, since we didn't have central reading in this study, in the phase 3 study, we have central reading of the endoscopies. So in this case, it was also good that we had it in one clinic. So it was one endoscopist who did all the endoscopies in this study as well.
Thanks a lot, Eva. And with that said, we open up for general questions, and there's a couple of questions coming in here. First of all, I don't know whether it's Jenny or Eva, but you released some pretty good. You released a positive press release last year from Japan. Are these still valid, and can you include that in the second part, in the study 2 of your phase 3 study, or is anything changed to that?
So the press release last year was very positive feedback from the PMDA that we would have the possibility to include Japanese patients in the conclude program without doing prior specific local Japanese studies. So that was quite positive. That's still the case. They haven't changed their mind. Whether we do include Japanese patients or not in induction study 2 is not decided upon yet because we haven't started that. But we definitely still have the option, and the go ahead from PMDA is definitely still valid. So no changes there.
Yeah, thanks a lot, Jenny.
And maybe you can also say that this pharmacokinetic study that we just did in this patient group was then important also for the discussions with the Japanese authorities, PMDA.
Yeah, thanks a lot, Eva. Yeah, and yes, so the only changes actually is related to the statement you came out with in January, as far as I know.
That's correct. Yes.
Yeah, good. And then, Jenny, if I may, there's a couple of questions about the market and the market growth. I know it's way before you entered the company, But do you still see the same growth rates, single digits to low double-digit growth for the overall UC market?
Yes, so we still see the same. The prevalence of UC is increasing in the world. So the number of patients is increasing, and historically, there's been a 10% market increase in UC. What we have said is that we believe more to be 6 in the future.
Yeah.
And you might ask why that is, since the patient prevalence is increasing, but that's because we account on prices decreasing. Okay. With more biosimilars coming into the market, so that will lower the value of the market, even though the patient and the prevalences are increasing.
Yes, and then there's a question here about the DIMS. I don't know if we should take that today or take it another time.
I think we can save it for another time. There's nothing new to report on that.
No.
So
good. I think, let's see if there's some other questions come up here. Yeah, there's one more here. No, that's about Japan. We already addressed that. We took the overall markets. Don't know if there's any other questions. Please post your questions now, and I will hand them over to management. Doesn't seem like that's the case. So with that said, I will close the session by saying thanks a lot, Jenny, for participating, and for you as well, Eva. Really nice to host both of you and to give answers to all the questions from the audience.
Thank you for having us.
You're welcome.
Thank you for having us.
You're welcome, Eva. And for all the participants, thanks for all the good questions. And with that said, enjoy your afternoon, and thanks a lot for participating.